Hi guys, today I want to go over a little bit of information to supplement what you've read about membrane transport. Most of the things, diffusion, facilitated transport, active transport, hopefully those make a lot of sense. But the way the water moves in osmosis and how that can impact the shape and the life of a cell can get a little bit complicated, and it's really important. Same as water is such an important molecule, what it does to our cells is really important too. So I want to spend a few minutes discussing that. The PowerPoint that I'm going to use is one of the PowerPoints that goes with your textbook, and you're going to see pretty soon why I don't use these very often. But I have put this PowerPoint on Edline, so you can look at the whole thing, but I'm just going to focus on one section of it for today. All right, so we want to talk about osmosis and how water in particular is going to impact the shape and structure of a cell and help maintain the water balance throughout your entire body. Okay, so remember that osmosis is specifically the diffusion of water across a membrane. Okay, so diffusion moving from areas of high concentration to low. Well, osmosis, water is so important that we give it its own name. The diffusion of water gets its own name, osmosis, but diffusion can happen through the air. Okay, as materials that you can smell move from their point of origin all the way over to your nose, that's still kind of diffusion, versus osmosis has to happen through a membrane. Okay, and the direction that water is going to move is going to depend usually on solute concentration. Water is usually going to move because some other material can't move. So for example, we've got this U-tube set up. Okay, and there's a selectively permeable membrane here in the middle. It's permeable to water, but it's not permeable to sugar. So the sugar molecules can't move through. If they could, they would. They would move through and balance out, but they can't. Every time they come up to this membrane, they bounce off. Can't get through. So, but the two sides here are not in balance. So the thing that can move is the water, and the water will move to make a balance. So since over here, there's all these solutes, that means there's less water. So water moves from where it's higher concentrated to where it's lower concentrated, and you can see this is where the balance happens. Okay? Water balance is all about concentration. It's not about number of molecules. Notice that the number of sugar molecules on each side of the U-tube did not change, but their concentrations are now even because now there's more water over here, so that lowers this concentration until it's even here. Okay, the water will move back and forth until it's even. Now remember, just because equilibrium or evenness is reached, that doesn't mean that the water just stops moving, now it's all frozen. It's still going to keep moving, but it's going to move so that one goes out, one goes in, one goes out, one goes in. Okay, so an overall net movement of 0, but there will still be movement. Okay, there's some terminology that we need to really pay attention to, and you're going to have to really remember for this section, okay? 3 words that you want to know: isotonic, hypertonic, hypotonic. And these 3 words refer to solute concentration. An isotonic solution is one that has the same concentration in point A and point B. Okay? Usually with this, we're talking about inside the cell and outside of the cell. So when a cell is in an isotonic solution, it's going to have the same solute concentration inside the cell as there is outside. There'll be no net movement of water, but water molecules will still move in and out at an equal rate. Okay? A hypertonic solution is one where there are greater. Hyper means more or greater. So whichever solution is hypertonic is going to be the area where there are more solutes. Isotonic, hypertonic, hypotonic, these are all terms of comparison. For example, you can't say that someone is taller. Oh, that guy, he's taller. Taller than what? Taller than who? You could say he's tall. Tall is an actual term, but you can't say that he's taller. A person can't be taller unless someone else is shorter. Okay? So one solution can't be hypertonic unless something else is hypotonic. So these are words that have to go together. They're comparisons. So you always want to make sure that you're paying attention. When you hear that something is hypertonic, what are they referring to? Which area are they referring to? Because whichever area they're referring to, the other area is hypotonic, has less. So hyper means more solutes. Hypo means less solutes. Okay? All right, so for example, here's some cells responding to changes in water concentration. So here's a paramecium. It's a single-celled organism. Okay, they live in fresh water. So they're constantly absorbing water into their systems because of the fact that they are hypertonic to their environment. Or the other way you could phrase it is that they live in a hypotonic environment. So their environment has less solutes than their body. So the environment is hypotonic and they themselves are hypertonic. Okay? Water is always going to move from an area that is hypotonic, has less solutes, more water, to an area that is hypertonic, more solutes, less water. Okay? So water is constantly moving into these guys. So they have this really cool structure called a contractile vacuole that can contract and spit water back out. So they're constantly spitting water out so they don't fill up too much with water. Because remember, non-plant and bacteria and fungal cells do not have a wall. If they do not have a wall, then that means that their membrane can rupture. So if a cell fills with too much water and it doesn't have a wall, it can rupture. All right, so let's look a little bit at this. So here's some different types of cells and different types of solutions, okay? So an animal cell, when placed into a hypotonic solution, water is going to move from hypo to hyper. It's going to move into the cell. If it keeps moving in and keeps moving in, because there's nothing to support or act as a barrier on the cell is going to explode, okay? A plant cell, however, needs to live in a hypotonic environment. That means that water is always moving into the cell. The vacuole, the main central vacuole of that plant cell is always full, and it's always providing pressure against the outside, against the wall of the cell. Okay, and that helps keep the plant rigid and upright. Remember that plants don't have bones. Okay, so they need something to help them stay upright. And this turgidity, or having their cells be turgid, tight, tense, helps keep the plant upright. Okay, if an animal cell is placed into an isotonic solution, it will look normal. Here's your red blood cell looking quite normal. Water's moving in, water's moving out at equal rates, all is well. A plant cell, on the other hand, when it's in an isotonic solution, the vacuole is going to shrink a little bit. And that means there won't be that rigidity or that turgidity. That's kind of a really fun word, turgidity. Won't be there anymore, and the cells are going to become what's called flaccid, and the whole plant is going to wilt. So when a plant wilts, it's in an isotonic solution. You need to give it more water so that there's more water outside than inside, so water's always moving into the plant. Now, if you put an animal cell into a hypertonic environment, water is going to leave the cell quickly, moving from the inside of the cell then is hypotonic to the environment, so water is going to move from the cell out, and the cell is going to shrivel in. Okay, it's why if you ever eat a whole bunch of really, really salty popcorn, you can feel like the edge of your lip gets all shriveled and wrinkly, and it's because all the water has left the cells in your lip, and it can make them feel really you can almost feel them shrivel up. Okay, now a plant cell has a wall. The wall is not moving. Okay, but if a plant cell is placed into a hypertonic environment, what is going to shrivel is the membrane. So the inner membrane is going to pull away from the walls of the cell, and everything on the inside is all going to shrink in. This is called plasmolysis. And a plant cell has become plasmolyzed when the whole plant does. I mean, it's like dry and completely shriveled up. It's almost past the point of saving. Okay, you would need somebody who's really good with plants to kind of nurse it back to health. Most of my plants end up this way. I kill them all. I try not to, but it always happens. Okay, so this is how water transport works and is why it's so important. I want to show you a little animation about this so you can get a little bit of a better idea about how this works.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis.
Side note for you real quick. Water can move directly through the plasma membrane because it's a small molecule. Even though it's really, really polar and the membrane is really, really non-polar, since it's such a small molecule, it can move directly through the membrane. It moves very slowly, though. So one thing that they never really bring up is the fact that the membrane has transport proteins in it called aquaporins, water holes. And aquaporins allow water to move through the membrane faster. Okay? Okay, so it can diffuse directly through the membrane, but it doesn't usually. It's usually moving through these special transport proteins called aquaporins. Just something to remember.
If a membrane is permeable to water, what happens when a solute cannot pass through the membrane? Experiment by dragging glucose molecules inside the cell, outside the cell, or both. Try different concentrations of glucose on each side. Before you run each experiment, predict which way water will diffuse and what will happen to the cell.
All right, so what we're going to do is we're going to take these glucose molecules, large polar molecules that can't diffuse directly through the cell, we're going to place them all outside the cell. All of them. So, now that there's all these solutes outside, what type of environment have I created for the cell? Hopefully you're able to guess that it's a hypertonic environment. The environment now has more solutes in it than the cell, so now the environment is hypertonic. So let's see what happens.
The cell is in a hypertonic solution. The solute molecules attract water molecules so that fewer water molecules are free to diffuse across the membrane. Thus, the concentration of free water molecules is higher on the inside of the cell than the outside. Osmosis occurs as water molecules move down their concentration gradient, leaving the cell. As a result, the cell shrinks.
So remember, it's animal cells that would do the shrinking. Okay? Plant cells are going to lose water too, but the actual physical size of the cell will not shrink. The inside will pull away from the wall. But an animal cell will shrink in a hypertonic environment. Okay, now let's do it the other way. Let's put all the glucose inside. So now what kind of environment is the cell in? Well, now there's less solutes out here than there are inside, so the environment is now hypotonic.
The cell is in a hypotonic solution. The solute molecules attract water molecules so that fewer water molecules are free to diffuse across the membrane. Thus, the concentration of free water molecules is higher on the outside of the cell than the inside. Osmosis occurs as water molecules move down their concentration gradient, entering the cell. As a result, the cell swells.
Okay, and again, it's an animal cell that will actually change its size. If you place it in a hypotonic environment for a little while, it'll just swell some. If you leave it in a hypotonic environment indefinitely, it will swell and burst. All right, so now one more. Let's put 2 on the outside and 2 on the inside. Now, unfortunately, because of the simplicity of this program, they're going to consider that even. And when it's even, what do we call it? Isotonic, hopefully you remembered. Now obviously, since a cell is much smaller than its environment, 2 molecules inside is going to have a much higher concentration than 2 molecules outside. So you got to remember that this is about concentration, not number of molecules, but they're simple, so we'll go with it for now.
The cell is in an isotonic solution. The solute molecules attract water molecules. Since the concentration of solute molecules is equal on both sides of the membrane, the concentration of free water molecules is also the same on both sides. Water flows back and forth across the membrane in equal amounts, and so the cell neither shrinks nor swells.
All right, good. Now we can look.
Click on the cells to see the effects of hypotonic environments on plant and animal cells.
Actually, I'm not going to do that. The one other thing that I do want to show you is I want to show you this video of plasmolysis. So this is when water is leaving a plant cell. And you're going to see your actual slides of this, but I want you to see the process in speeded up time lapse. Okay, so here it's playing, and you can see each brick is its own cell. So focus on just this brick right here. Let's see what happens. It happens really slowly, so you might not really be noticing anything right now. Okay, now that it's done, you see this brick right here? I'm going to take it all the way back to the beginning, and I want you to notice the change, what has happened. Okay, notice what that brick looks like there. And now, start it again from the beginning. Look at that. See how here it's a constant mass. You can see all the cytoplasm, all these green chloroplasts spread out evenly throughout the entire brick of the cell. Okay, that's what the cell should look like. By the way, this is an allodious cell, which is a type of algae. Okay, now I'm going to do this really fast at the end, and you can see what it looks like when it goes really fast, and you can see it kind of pull in. Look at that. Pulling, 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 pulling. There you go. There's the plasmolysis, all the way so that membrane has shrunk all the way into the beginning. Regular cell, plasmolyzed cell. All right, guys, have a good day.